We'll just have a minute until our next speaker. She is a research scientist and joint faculty here at the University of Missouri and a research scientist with the USDA ARS, Dr. Kristen Veum. So thank you, Kristen. Welcome.
All right, so I guess so first I'd like to thank Gregory for the invitation. It's not very often that I get a chance to really dive into some of the details about what I do and talk about methodology and tests for producers. But to give you a disclaimer, I'm a lab scientist. I've been in the lab in one form or another for almost 30 years. So I started back in high school. So if you want to do the math and figure out how old I am, that's up to you. But I'm going to talk about a specific aspect of my research, which is developing affordable and interpretable tests for producers. So how many of you are farmers or producers? Okay, so what I'm doing here, this work is for you. And how many of you are lab people? I know I see some out there. Yes, Mangela, raise your hand. Okay. So soil health is about that intersection of these ecosystem services. We've heard a lot about that today. And it's about trying to simultaneously optimize all of these ecosystem services. So as a lab scientist, then, my job is to find soil measurements that are chemical, physical, and biological that reflect these ecosystem services. So this is just an example of some of the measurements we've used in the past. This was from one study, and obviously there's an awful lot going on here. So the question then becomes, what do we measure, and how do we measure it? And what are really the important things that we want to measure? So these are the steps that you think through when you're trying to develop something that's going to be agronomically meaningful in terms of a soil health assessment for producers. So the first step then is to select those soil functions or those ecosystem services that you want to represent. Then you go on to select the indicators or the measurements that reflect those functions, right? And then you have to select a laboratory method. And this might seem very straightforward or simple, but it's really not, because we don't have standardized methods in place for a lot of these soil health indicators. And then number 4, I put that in red because that is really the hard work of a soil health assessment. And that's quantifying that relationship between these indicators that you're measuring and those soil functions or those ecosystem services. And the 5th thing then is to quantify the effects of management on those indicators and functions so we can understand the role of management in soil health and ultimately to be able to make recommendations to producers. So this is a list of characteristics of soil health indicators that I think are important from the context of producer testing. So if we're going to measure something, we want it to be meaningful and reflect those soil functions. Okay, so to be meaningful, that can be scientific, agronomic, or have environmental relevance. Then we got to think about the sensitivity of what we're measuring. So we want to optimize that sensitivity. We want it to reflect, ideally, some short-term changes, something under 10 years, right? So if you're a producer, you're making a management change, you'd like to have some way of monitoring the effects of that on the near term, right? But then we can also measure things like organic matter or total carbon that might be a little slower to respond. The flip side of that, of course, is you don't want it to be too sensitive. So if it's hypersensitive, it's high variability, or changes a lot just based on day-to-day weather conditions, then it's probably not a very good indicator. And we want them to be measurable. And what does that mean? Well, we want standardized field and lab protocols. And that is ultimately so that we can measure something that's repeatable and reproducible. This 4th thing here, this comes back a lot to the fact that we're talking about producer testing. It needs to be affordable. So you want it to involve minimal equipment, consumables, labor, and skill if possible. And then again, I'm going to keep coming back to this last point. We want it to be interpretable. And so that, again, comes back to it being scientifically based and provide decision tools for management. So that's the agronomic and the scientific components. So when we go through method selection for a producer test, this is going to be a very different process than what you might do in a research lab. Or, if you're in the business of studying soil just for the sake of studying the soil, which I like to do that too, you might want to use some really high-end equipment and measure something that's very expensive or very exotic, right? But if we're going to come up with something that's going to be for producers and be accessible for service labs, then we go through a different selection process. So the first thing then is the time and cost of sample collection and preparation. So you're asking producers to go out and do this, right? They're going to collect these samples, or maybe they're working with consultants or agency representatives. But you don't want them to have to invest a lot of time and effort in doing that. And ideally, if you can pair that with other testing they're doing, like fertility testing, that would be great. In terms of the cost per analysis, you got to think about all the consumables and the labor that are involved in running that analysis in a lab, and capital equipment costs. So if you have to buy a $200,000 mass spec, is that really realistic for producer testing? And then the next point, the required skill level to run that instrumentation. Now we're really lucky here in Missouri. We've got the shack, we have Russ and Donna. They do some pretty high-tech things there. But a lot of labs don't have access to that level of skill. So if you buy that $200,000 mass spec, then you're going to have to also have highly skilled and probably highly paid personnel to operate it. And finally, all of those costs have to be balanced with interpretability. So a lot of the research in soil health right now is focused on the soil biology, and in particular, the soil microbiology. And one of the many reasons to focus on the microbiology of the system is that you can collect a soil sample and send it off to a lab, and you can look at the soil microbes, whereas some of the macrofauna or the mesofauna, you can't handle in that way. So what do we do if we want to study the soil microbes in the lab? So we can look at how many microbes there are in the soil. We can, and you can think of that in terms of like a population or a census. You can measure the biomass and the total phospholipid fatty acids. But you probably want to drill down a little deeper into that and see what's the microbial community structure. So you want to know what kinds of organisms are there, those different groups of organisms. And you can do that by looking at microbial DNA, or again, you can go back to the phospholipid fatty acids. On the DNA side, I don't have any data to show you yet, but we have multiple studies ongoing. We have a collaborative project with ARS in Florence, South Carolina, looking at the microbial community under cotton systems with different conservation management. We have another project working on our local cropping systems, and we're collaborating with BYU to do the microbial genetics part of that project. And then my postdoc, Chen-Wei Li, raise your hand, is also looking at prairie restoration and the effects of simulated drought on reconstructed prairie. And we'll be sending our samples to Argonne National Labs for the sequencing in that project. When it comes to the microbial PLFA analysis, I have so many projects that we have PLFA data that we've primarily sent all those samples to the shack since they geared up back in 2012. And so we have a ton of projects and data. And so this, on your bottom left, is an example of what some of our PLFA results look like. And that's looking at the effects of management practices on total PLFA, that's the height of the bar, as well as the distribution of the microbes in those different groups. So the last question here, what are those microbes doing? This is the one that's really the most interesting to me, because you can sequence the genomic DNA of the microbial community, and that can tell you what the genetic potential of those organisms are, but it doesn't really tell you what they're doing. So we have a variety of ways we try to go in and figure out what those guys are really up to. Microbial enzyme activity is one I focus on a lot. There are a whole host of enzymes that we can measure that reflect nutrient cycling in the system, and other things like soil respiration. It tells you about the activity of those microbes. But ultimately, the big challenge that we're facing right now is linking that microbial information to those soil functions and those ecosystem services. Because we need this to be outcome-based, and again, we need that agronomically meaningful interpretation. There's a lot of interest in this area of research right now. There's a lot of money going into soil health research in general, and specifically studying soil biology and soil biological measurements and figuring out how we can interpret that data. So, as I mentioned, a lot of these biological measurements are expensive, at least currently. Costs are coming down for a lot of them. But we're also working on ways to inherently reduce the cost of testing these things. So one of my ARS colleagues, Veronica Acosta-Martinez in Lubbock, Texas, has developed a very new, this is like hot off the presses, multi-enzyme or simultaneous enzyme test, where you can, with one sample, measure anywhere from 2 to even 4 enzymes at the same time. And by doing that, now you can capture multiple nutrient cycles simultaneously. And right there, you've drastically cut the cost of doing that assay. Another test that's showing a lot of promise is soil respiration. You've probably heard about that. There are probably 10 or 20 different ways to measure soil respiration. But the one that I'm looking at right now that I like is Cornell's, what I call poor man's respiration. So I don't know if Harold Van Ness would really appreciate that, but that's what I call it, because you can see in the picture it just requires a mason jar, a little aluminum cup, and that's a pizza table underneath that tiny little beaker, right? That little thing that comes in your pizza. But anyway, this test is showing a lot of promise, and you don't need an expensive GC, you don't need expensive equipment to generate that data. Another thing we're doing is, again with Veronica Acosta-Martinez in Texas, we're comparing that phospholipid fatty acid data with ester-linked Veum data to see if we can get comparable information on the soil microbial community, because that particular assay, at least as of right now, is about 50 to 60% less expensive. And then we're also always looking at ways to use smaller sample sizes and smaller volumes to cut back on things, turn something into a microplate method to make it more high throughput and speed it up. And then coming up with ways, methods where you can use dry soil samples so you don't have to worry so much about the time of sampling and the shipping costs and overnighting samples to your lab. And again, ultimately, we want to balance the information that you're getting from these tests and the value of these tests with the cost of doing them. So after we get all this data, these chemical, physical, and biological data, then what do we do with it? How do we interpret it? And the basic approach comes down to using one of many different soil health indices that are out there. But the process is as shown here. You collect your data, that's your laboratory data. You put it through some function, some interpretation or scoring function. And then on the end, at the output from that then is a score that is more easily interpreted. So there are some characteristics that, and this is my list, this is my perspective on using a soil health index. So this is biased by my perspective, but a good soil health index is going to account for site-specific characteristics. I think that's the first and foremost consideration that you have to keep in mind. Those inherent soil and climate characteristics. And I'll talk a little bit more about that. It also needs to include chemical, physical, and biological measurements. You can't just look at the biology or just the chemistry or just the physics. You need to bring it all together for it to be really a comprehensive soil health assessment. And this includes soil fertility. So fertility is part of soil health. We're not throwing the baby out with the bathwater. Okay, we're expanding upon that, and fertility is an inherent part of a good soil health assessment. And then again, ultimately what we want to be able to achieve is providing a meaningful interpretation based on these agronomic and environmental thresholds. And why do we want to do this? Well, there are a lot of reasons, in a lot of ways we could use this information, but we can rapidly identify where and how a soil is not performing and that aids in prioritizing management decisions. So I'm going to give you guys a little quiz. Are you ready? Okay, who can tell me what Hans Jenny's 5 soil forming factors are? I know some of you have had soils classes. Got to dig back in time here. I see, so come on, somebody throw them out.
You just said time.
Yes. So time is one. Parent material. All right, what else? Climate, yes.
Acting on topography over time.
Okay, Wayne, thank you. Thank you. All right, we have a winner. Okay, so these are the 5 soil forming factors. And these are the things that go into producing the soils as they exist today. Oh, and can anyone tell me, and this was in Randy's pedology class, we talked about the 6th soil forming factor. Anybody tell me what that one is?
Would it be humans?
Yes, right again. Okay. So you didn't even take its class. I know lots of you out there did. So that's management, or the human factor, right? So all these factors that go into forming soils in their inherent state, but then we come along as humans and we manage it and we do other things to it and we change its state. So I'll talk just briefly about some of these things. Climate. This is a climate map of the United States. And I think it's pretty obvious the link between climate and agriculture and ultimately soil health. And we have quite a gradient of climate here. And that's a major factor in the types of soils that we have across the country. And so here's a map of the major soil orders. Does anybody want to tell me what those major soil orders are here in Missouri? What are the big ones? I'm listening. Alpha soils, that's one, yes. Mollisols. Yeah, so okay. All right. So you guys are still awake, I think. All right, so, but notice that big green area in the middle of the country, right? So who can tell me what this is a map of? Prairie. Okay, I got lots of answers there. That's right. So do you see any similarities between the map of the prairie and the soils? Do they look at all the same? And do you think that's coincidental? You know, there's a relationship between soils and vegetation, right? And then if we throw in a map of cropland acres here in the US, it looks a lot like that prairie map, and again, like that soils map. So again, that's the role of soils and vegetation in this whole process. So why do we care then about soil taxonomy? Well, you know, I'm not going to go into all the details here, but we know soils are not all the same. Mollisols are not the same as alpha sols, and one of the key components of soil taxonomy is that texture. And it's the soil texture that's going to determine how much organic matter that soil is even capable of retaining or building. It's also a key component in how much water the soil can hold. And I think Wayne had some slides related to that in his talk. So if you put this all together, this is my diagram or my representation of soil health. So you start with the pedology, those 5 soil forming factors. And that defines really the extent of what's possible with a soil. What is that inherent potential of any given soil? And then you throw in the management component, and then you start diverging into any number of different directions, and then we have this gradation of soil health. So there are several different soil health indices out there that are available to use. There's the Soil Management Assessment Framework. This is the one that I like to use the most, because it does utilize soil taxonomy and a variety of site characteristics and these nonlinear scoring curves. I have on there that it may need more regional validation. I should just cross out the word may. It definitely needs more regional validation. That's one of the things that we're working on with other ARS scientists right now. There's also the Comprehensive Assessment of Soil Health. That's the CASH, that's Cornell's system. They originally based that on soils from New York, but they have more recently brought in Midwestern soils and modified their algorithms. There's also the Haney test, and then some non-agricultural systems out there for forest soils, wetland soils, and prairie soils. And And even though we may think of those as being as natural systems, I think you're pretty hard-pressed these days to find any system that's not affected in some way by humans and by management. And even our remnant prairie here, east of town, it's managed through prescribed burns and other methods. So even in those ecosystems, we want to think about how management affects soil health. So like I said, I use this MAF, and it checks off most of those criteria that I had for a good soil health index. So again, it has the chemical, physical, and biological components. It also includes fertility, because for producers, you really need that information. And you can see there's a range of different measurements that you can choose from. But it accounts for soil taxonomy, texture, slope, and climate. And it's the only soil health index that really made an effort, going back to when it was first developed, to do that. So I know I'm on a university campus here. I'm talking to a bunch of scientists and producers. I felt it was important to at least have one slide with a bunch of equations on it, right? So you could feel like you're getting your money's worth today. So these are the algorithms, at least some of them, that the SMAF uses to parameterize those scoring curves. So I'm showing you pH. That's an optimal curve. And a lot of that is parameterized based on the crop. It's very crop-centric. If you look at PMN, it's dependent on organic matter class, texture, and climate. Phosphorus, this is very unique, and it's a good thing to point out here, is that the ascending curve of the phosphorus algorithm is based on plant needs, so the crop requirements for phosphorus. However, if you have excess phosphorus and you combine that with slope, then you get penalized in your score. And then we have beta-glucosidase, and so anyway, there are algorithms for every indicator in this MAF. So this is an example of where I use this MAF on our Centralia research sites north of town. We had a variety of corn soybean systems, some CRP, and some on-farm, adjacent on-farm sites. And we measured all those different things. We put them in this MAF. We scored it. And if you see, there was a general divide between the perennial and the annual cropping systems. Within the perennial systems, we saw effects that were based on the type of vegetation that was present. And then within the annual cropping systems, we found that tillage and rotation diversity were the primary factors. And again, we did this at Sanborn Field. This is a site that we sampled it. At the time, it was, I think, around its 120-year anniversary. It's now at 130 years that it's been in operation by MU Extension, has been keeping this long-term site going. And it's a perfect example of soil health research. And we saw very similar results when we looked at the SMAF scores here. So again, we saw that split between perennial and annual cropping systems. And then within the annual cropping systems, in this case, we're looking more at rotation and amendment practices. So I put all this together in what I called my agricultural continuum of soil health, based on perennial versus annual, and then all the different management choices that are made within those types of cropping systems. However, it's limited to those systems that I studied. So one of the things I'd like to see in the future is that we expand research into more diverse and alternative management systems. So right now, FAR has an initiative to put research money into rangelands and pasture. And that includes livestock and grazing practices and things like grazing cover crops. And I've done a little bit of work in this area with Todd Lorenz of MU Extension. We had some grazing and pasture work that we've done recently. But also organic systems and vegetable systems. Someone mentioned earlier, within organic systems, there's a lot of tillage used for weed control. So people like Tim Reinbott and Carrie Clark are looking at ways to try alternative management approaches so that we can reduce the soil disturbance but still maintain the integrity of the organic system. We also need more work in forest and prairie systems, partly to understand those ecosystems, but also to understand what are the effects of burns and harvest and clear cutting activities. And ultimately, I'd like to see more studies of mixed perennial and annual cropping systems. Things like the agroforestry systems, like the work that Ranjit Udawada has been doing. So we can understand when you bring together different types of vegetation, what effects and what benefits that has for soil health. So where are we now in terms of regional and national research? We've identified these important soil functions. We've selected indicators. We have methods and protocols. But what we really need now is that regional interpretation for management decisions. So we need to bring together that data. We need to merge the soils data with the water quality data, with the crop productivity data, and start looking at how those things all come together. And if I take you back in time briefly to the 1990s, these were all the arguments against the concept of soil health. And a lot of them were very well put and very well posed. But I think since then we've made a lot of strides towards resolving and answering some of these questions. But we're still working on the interpretation. And the take-home points are that we need to study more diverse and alternative systems. And I think if we're going to interpret soil health, we need to do that on a regional basis, on different scales, depending on the main factors that are influencing that. And then ultimately, we need to focus on developing those producer recommendations that are meaningful, decision-based, and cost-effective. So that's it. Thank you.
Thank you, Kristen. Thanks for that excellent presentation. We do have time for a few questions. Dr. Lloyd Summer, please.
Very nice presentation. I appreciate that.
Thank you.
Many people, when they talk about the chemistry of soils, just focus on nitrogen, phosphorus, and potassium. But we are beginning to understand that the chemistry is much more complex than that.
Right.
And how these compounds are speciated accesses their or influences their bioavailability. So my question is really how much further do we need to dig into the chemistry of soils to better understand this? You gave an example. Many people just do inorganic phosphates, but you did phospholipids, which is another pool of phosphorus that can be accessed.
Right.
So again, the question is, how much further do we need to dig into the chemistry and biochemistry of soils to really understand that influence on soil health and plant growth and productivity?
Right. So I think in large part our knowledge in these areas is very compartmentalized. So a lot of people have studied the nitrogen cycle. We know those different nitrogen species. We understand the transformation from organic nitrogen to inorganic nitrogen and plant available nitrogen. But what we don't necessarily understand is how it all comes together in the context of the soil. So how that interaction, and in the ARS we now have this initiative, genetics by environment by management interactions. And so it's pulling these things together and understand how they relate to each other so that we're not studying just one component in isolation. I don't know if that answers your question, but.
Are there any pools of chemicals that we need to be looking at in the future? So again, the question was, are there any additional specialized pools of biochemicals that we need to be looking at? For example, you gave phospholipids. Are there other nitrogen compounds?
Well, when it comes to nitrogen, right, you can look at the proteins, which are primarily microbially derived in the soil. But it's, you have this organic nitrogen pool as a whole that's available potentially to plants. And a lot of work has been done over several decades to try to measure that organic nitrogen and relate that back to crop productivity so that potentially we can reduce our nitrogen fertilizer inputs. However, there are all these interactions with climate and with timing. When is that nitrogen being mineralized by the microbes? Is it when the plant needs it? So there are a lot of things that, again, when we study it in isolation, maybe we can't answer that question. But science it's all very complex, and if we can do a better job of understanding that complexity, then we can figure these things out.
Okay, well, thank you very much. Thank you, Kristen, and for those questions.